everyone, welcome to Progressive Medical Laboratory Technology. That is the first class from our page as well as from our YouTube channel. From today we are starting a new journey that will be from every week you will get the video from our YouTube channel. Today is the first class of microbiology, so that here I am writing the class 1 and we will discuss about the microbiology today. Today is the introduction class, as well as today we will understand how do we learn the whole microbiology. See here, firstly micro define small, bio define organism, as well as logy define study. So that microbiology defining the study of microbes or study of the organism who are small. We cannot see the microorganism into our naked eye, so that whenever we are discussing about the microorganism, that is called microbiology. Now the second question is, who are and what are the microorganisms? Basically, there are 4 types of microorganism. First is bacteria. Whenever we are discussing about the bacteria, that is called bacteriology. Secondly, virus. When we are discussing about the virus, that is called virology. After that, we are discussing about the parasite, that is called parasitology. And last one is the fungus. Fungus means mycology. We are discussing the fungus in our mycology chapter as well as in our mycology section. That's all are the basic things about the microbiology. Whenever we are understanding the microbiology, we need to understand bacteriology, we need to understand virology, parasitology and mycology. That is the introduction class, so that I will give you only the touch up of the microbiology. And whenever we are starting the class one by one, you will get everything from our YouTube channel. Okay, see here I have written down medical microbiology. Though it is a channel of medical laboratory technology as well as the NEET aspirant as well as the medical student, so that here we need to understand the microbiology. Whenever we are discussing the microbiology in the aspect of diagnosis the disease, that is called medical microbiology. So that the M.L.T student or lab technology student basically gaining the knowledge of the microbiology, they all are the medical microbiology, because they are understanding the microbiology in the aspect of diagnosis disease. Understood? That's all are the very basic things. There are, firstly we will start the bacteriology. Bacteriology. Firstly, we will start the bacteriology. There are 2 types of microbiology. One is general microbiology we need to understand, and after completing the general microbiology, we will go to the systemic microbiology. See, general microbiology means we need to understand how the whole phenomena are occurring. And systemic microbiology, we will understand the application of general microbiology. Systemic microbiology, we will understand about the every type of bacteria one by one, who are basically belonging into various type of system, as well as who are basically causes various type of disease into our system. That is systemic microbiology. Firstly, we have to cover the general microbiology. After that, we will go to the systemic microbiology. Okay, see, firstly bacterial morphology. We need to understand how a bacteria look like. That is the morphology of bacteria. After that, how the bacteria growth is occurred, as well as which kind of nutrition is required for bacterial growth, we need to understand. Because without knowing the nutrition, we cannot grow the bacteria. As well as we will understand the growth curve in the section of growth and nutrition. After that, we have understood that which kinds of nutrition are required for the bacterial growth. Now we will understand which media as well as where basically we are growing these kinds of bacteria. That is called culture media. We will understand what are the types of culture media. We will understand what are the basic ingredients of this culture media. We will understand about the specific culture media which is required for every bacteria, as well as there have some kinds of selective enrichment culture media. We will understand they all are specific for the specific bacteria. We will understand this. After that, we will understand. After preparing the culture media, we will go to the culture techniques. Culture technique means how do you do the culture? That then maybe streaking, that may be spread culture, that may be portrait culture, that may be staff culture. There have lots of culture technique. We will understand it. After the see here, there have some kinds of biochemical test. We need to understand the biochemical assay. Means bio means there have the organisms, and chemical there should be reaction will be occurred. So that we are saying that biochemical reaction. And I am just giving you one or two example: indole, oxidase, coagulase, citrate utilization test, methyl rate test. There have lots of tests we will understand in the section of biochemical assay. After doing the biochemical assay and the media is grown, then we have to taking the culture, as well as we have to taking the taking the sample as well as the inoculated things, and we will do the staining. There have various type of staining we need to understand. There have some simple staining, there have some differentiated staining like gram staining, AFB staining, there have Albert staining, there have Indian ink staining. There have lots of type of staining we will understand in the section of staining. After that, last one is the antibiotic sensitivity test. We need to understand which kinds of antibiotic are sensitive, which kind of antibiotic are resistant for a particular bacteria. For a particular bacteria, we need to understand. That is called antibiotic sensitivity test. We will use this lots of number of antibiotic against one bacteria. Getting my point? After that, we will see is it sensitive, is it sensitive or resistant. Sensitive means it's working, resistant means it's not working. We will understand these things. So that's all are the general microbiology. We need to understand the general microbiology at first. After that, we will go to the systemic microbiology. Systemic microbiology, we will understand the whole bacteria one by one. Thank you so much for listening to this video up to this much. We will meet soon.